Welcome to this lecture on animal biotechnology. Today, we are going to explore a fascinating and highly advanced technique known as DNA microinjection. Imagine trying to insert a tiny rolled-up paper message inside a microscopic water balloon without popping it. This is exactly the challenge scientists face when they want to give an animal a completely new genetic trait. By the end of this session, you will understand exactly how this delicate microscopic surgery is performed and why it is so crucial for modern medical research. To begin our journey, let us understand the fundamental challenge of gene transfer. When scientists want to introduce a new genetic trait into an animal, they cannot just rub the DNA on the outside of the cell and hope it magically finds its way inside. The cell membrane is a strong barrier. Instead, they must physically force the genetic material in using incredibly precise microscopic tools. This process is very much like performing delicate surgery on a single fragile cell. By doing this successfully, we can introduce new instructions into the biological brain of the cell, permanently changing the future of that animal and all of its descendants. Now that we understand the challenge, let us look at how the actual procedure is performed. DNA microinjection is a highly advanced physical method of gene transfer. In this technique, a scientist uses a microscopic glass needle, which we call a micropipette, to pierce the outer membrane of a newly fertilized egg cell, known as a zygote. Because the cell is so incredibly small, this entire procedure must be performed under a powerful microscope. The scientist uses a robotic device called a micromanipulator. This brilliant piece of equipment translates the scientist's large hand movements into tiny microscopic needle movements, ensuring the cell is not destroyed during the injection. Building on the use of the micromanipulator, we must now ask, exactly where inside the cell does the DNA go? Once the needle is safely inside the zygote, the foreign DNA is not just released randomly. It is injected directly into the male pronucleus. The male pronucleus contains the sperm's genetic material just before it fully fuses with the egg's genetic material. Why target this specific area? Because placing the DNA directly into the pronucleus gives it a very high chance of integrating into the host's genome. This critical integration happens before the cell even begins to divide and grow into an embryo. Let us visualize everything we have discussed so far with this detailed diagram. On the left side of the screen, you can see a large, blunt instrument called the holding pipette. Its job is to apply gentle suction to keep the spherical zygote perfectly still. Inside the zygote, you can clearly see two distinct circles, the female pronucleus and the male pronucleus. Notice how the incredibly sharp microinjection needle enters from the right. It carefully pierces the outer membrane of the zygote and goes straight into the male pronucleus. The tiny dots you see exiting the needle represent the foreign DNA being delivered exactly where it needs to be for successful integration. Having seen the procedure, you might wonder why we go through such a difficult and tedious process instead of using easier chemical methods. The answer is that DNA microinjection is the absolute gold standard for creating transgenic mice. These animal models are absolutely essential for studying complex human diseases like cancer and Alzheimer's. Furthermore, unlike viral methods, microinjection does not require the use of potentially dangerous viruses to carry the DNA into the cell. It also allows scientists to inject very large pieces of DNA, which viruses simply cannot carry due to their limited physical size. This makes it an irreplaceable tool in medical research. Since this procedure is so delicate, we need a way to measure our success, which brings us to the transgenesis efficiency formula. Conceptually, this formula tells us what percentage of our starting injected embryos actually resulted in a successful transgenic animal. The formula is E equals N sub T divided by N sub I, all multiplied by 100. Here, E represents the overall transgenesis efficiency as a percentage. The symbol N sub T stands for the number of transgenic offspring that are successfully born and carry the new gene. The symbol N sub I represents the total number of fertilized embryos initially injected. If the number of successful transgenic offspring increases while the initial number of embryos stays the same, the efficiency goes up. Conversely, if many embryos die, the efficiency drops significantly.
Now that we have our formula, let us see this with a concrete example. A research laboratory injects a human diabetes gene into 400 fertilized mouse embryos. Later, 40 pups are born, but genetic testing shows only 8 actually carry the gene. We need to calculate the efficiency. We are given that the initial number of injected embryos is 400, so N sub I is 400. The number of successful transgenic pups is 8, so N sub T is 8. Notice we ignore the 40 total pups born because efficiency measures success from the very first step. We substitute 8 and 400 into our formula. Dividing 8 by 400 gives 0.02. Multiplying by 100 yields a final answer of 2%. Physically, this means for every 100 embryos injected, only 2 become transgenic mice, highlighting how challenging this process is. That low efficiency rate we just calculated leads us to another critical aspect of the procedure: precision. When performing this delicate procedure, scientists must control exactly how much DNA goes into the cell to avoid damaging it. If too much liquid or too many DNA copies are injected, it can easily become toxic and kill the fragile embryo. Therefore, the total number of genes delivered is carefully calculated based on how packed the genes are in the liquid and exactly how much liquid is squeezed out of the needle. This requires incredibly precise calibration of the microinjection equipment before the experiment even begins. To ensure the embryo survives, we use a simple but vital mathematical relationship. This formula dictates exactly how many DNA molecules are delivered into the pronucleus. The equation is N equals C times V. Here, the variable N represents the total number of DNA molecules, or copies, injected into the cell. The symbol C stands for the concentration of the DNA solution, typically measured in molecules per picoliter. Finally, the variable V represents the incredibly small volume of liquid injected, measured in picoliters. If the injection volume increases while the concentration remains constant, the total number of injected copies increases. If this number gets too high, it threatens the survival of the embryo. Let us apply this formula to a real laboratory preparation. A researcher needs to inject exactly 500 copies of a gene into sheep zygotes to get the best results without killing the embryo. The needle delivers a precise volume of 2 picoliters. We need to find the required concentration. We are given the target number of molecules, N, which is 500. The volume, V, is 2 picoliters. We start with our formula, N equals C times V. To isolate the unknown concentration, we rearrange the formula by dividing both sides by the volume, giving us C equals N divided by V.
Substituting our values, we divide 500 by 2. The final answer is 250 molecules per picoliter. Physically, the researcher must dilute their sample until exactly 250 copies exist in every single picoliter of liquid.
We have covered a lot of ground today, so let us summarize the key takeaways. First, DNA microinjection is a powerful, direct, and highly precise physical method for introducing foreign genes into a living organism. By injecting DNA directly into the male pronucleus of a fertilized egg, scientists can safely transfer large genetic sequences without relying on viral vectors. We also learned that while the procedure requires immense skill and suffers from relatively low overall efficiency, precise mathematical control over DNA concentration and volume makes it possible. Mastering this concept is absolutely essential for understanding how we create transgenic animals for medical research and pharmaceutical production. Thank you for joining this lecture.